Cell division is important in both animal and plant cells for growth, development, and maintenance. The cell cycle control system is key in mitosis. It is a cycling operating system of molecules in the cell that triggers and coordinates key events in cell cycle. Cell cycles have a checkpoint where stop and go ahead signals can regulate the cycle. Many animal cells show anchored dependence. The cell must be attached to the inside of a culture flask to even start division. The cell goes through mitosis deriving from binary fission because eukaryotic cells had nuclear envelopes but larger genomes evolved. Mitosis has 5 stages: prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. During prophase, chromosomes condense and the nucleolus disappears. The mitotic spindle is also forming. During prometaphase, chromosomes are visible and they have 2 sister chromatids and the nuclear envelope will fragment. During metaphase, the spindle is finished and the chromosomes that are attached to the microtubules are all at the metaphase plate. During anaphase, the chromatids are separated and the daughter chromatids are moving to the ends of the cells as the microtubules shorten. During telophase, daughter nuclei are now forming, and during cytokinesis, the cell plate divides and is growing towards the perimeter of the parent cell. Only diploid cells go through meiosis, and chromosomes and diploid cells are duplicated, and the copies are sorted into 4 haploid daughter cells. And even though there's 1 duplication, there's 2 cell divisions. Together, sister chromatids make up 1 duplicated chromosome. In prophase 1, duplicated homologous chromosomes pair in extreme segments. In prophase 2, a spindle apparatus forms and chromosomes move towards the metaphase 2 plate. During metaphase 1, chromosomes line up by homologous pairs, and in metaphase 2, the 2 sister chromatids are not genetically identical because of the crossing over in meiosis 1. The sister chromatids have an attachment to the microtubule extending from opposite poles. The G1 checkpoint is where the eukaryotic cell decides whether it wants to divide or not. Once it enters the checkpoint and enters the S phase, then it has to divide. Even if there are damages in the DNA, the cell will still go through the G1 checkpoint and produce 2 daughter cells. The G2 checkpoint checks to see if the DNA is damaged and if the DNA was completely copied in the S phase. If there's damage, the G2 checkpoint will stop the cell to fix it, but if the cell is irreparable, then it goes through apoptosis. Apoptosis is a programmed death of the cell. This helps prevent cancer cells from passing through. The M checkpoint is to make sure the daughter chromatids are correctly attached to the microtubules. The cycle will not go through until all chromatids are firmly attached. If a chromosome is misplaced, then mitosis will stop so that microtubules can catch all the lagging chromosomes. The internal-external signal cell if the cell is large enough, if the cell has enough energy reserve, and if the cell is picking up on positive cues and the DNA is damaged. Cyclins are the most important cell regulators. They cause DNA replication and chromosomal separation. Cyclins perform these steps with a group of enzymes called cyclin-dependent kinase. CDK are enzymes that attach to a phosphate group in specific proteins. Cyclin and CDK work together to control cell cycle transitions called maturation promoting factor. The MPF complexes add phosphate tags to several different proteins in the nuclear envelope, resulting in its breakdown. Platelet-derived growth factor signal critically regulates post-infection repair. Cancer is a disease of uncontrolled cell division. Its development and progression are usually linked to a series of changes in the activity of the cell cycle regulator. Cancer cells may make their own growth factors, have growth factor pathways that are stuck in the on position, or in the context of the body, even trick neighboring cells into producing growth factors to sustain them.